So chapter 17 is selection and evolution. So when we come to chapter 17, remember, chapter 17, we will always refer to a population. Whatever that we say inside this chapter, we always refer to a population. Within the population, selection can happen, and within the population, evolution can happen. So it's with reference to a population already, not an individual. Okay, so let's look at the learning outcome for this lesson. The first topic is variation. Now, this variation is very familiar to all of you, because if you look around your classroom, why is it you know how to identify Lim Yi Hao, okay, Megan, Curti, Januli, Andrea, because they look different, right? So this is called variation, right? Variation. So we're going to learn about variation in this lesson. Now, whatever feature that you can see in this person, right, that allows you to identify them is called the phenotype. Okay, phenotype. So phenotypic variation, that means the differences in all of you that allow you to identify each other is because of genetic factors. That means the genes and alleles that you inherit from both your parents, and plus on top of that one, environmental factors. Environmental factors means what kind of food you eat, what kind of lifestyle you have, what kind of stress level you have. Okay, whether it's holiday time or exam time, the person look different. Okay, so this is called environmental factor. So therefore, VP is equals to VG plus VE, where V stands for variation. So the differences in the phenotype in your class is because of the differences in genotype plus the differences in their environmental factor. So this is the first thing we're going to learn today. After that, we're going to look at two types of variation, which is discontinuous and continuous variation in the phenotype. Okay, and then after that, we're going to examine their genotypes, the genetic basis of these two variations, which is discontinuous and continuous variation. Okay, so let's look at the variation. Now, for the learning outcome, got t-test, but I've taught you t-test already, right? No, I'm going to teach you some other day. Okay, t-test is also in this chapter. That means this t-test can come out in paper 4 also, right? Other than paper 5. Okay, all right. So you need to fill in the empty table inside your notes. Your notes is empty one. Okay, so genotype plus environmental factor will give the person the final phenotype. Because genotype is nature. Nature means you're born with it in your genes and alleles. Environment will nurture your genes to give the full potential. So we will take these 4 examples to illustrate this. Okay. Human height, Himalayan rabbit fur color, corp length, and skin color and lactose metabolism. Okay. So first example is human height. Now, human height, you got a gene for human height. Plus, if the person is malnutrition, then the person will not be so tall, so be short, isn't it? Even though you got the gene for height. Because you need proteins and calcium to build the bones in order to be tall. Okay. If a human height gene is tall plus sufficient calcium, then this person will be tall. So therefore, the final phenotype that we can see with your eyes is not only because of the genotype, but also because of environmental factor. That's why nature plus nurture. So the allele will code for the maximum extent of individual's height. How tall can you grow? But in actual fact, how tall you grow depends on environmental factor. It means the nutrients that you're able to obtain from the nature to support the expression of the gene to its maximum potential. Second example, Himalayan rabbit fur color at the tips. This one, I have to show the diagram first. Now, this Himalayan rabbit, right? They have alleles to code for dark pigment, but the dark pigment will only be switched on. The allele will switch on only in low temperature. So when it's very cold, only those genes are switched on to give dark fur. When it's higher temperature, those genes for dark pigment will not be switched on, so it's light fur. So the environmental factor here will determine what allele, what gene will be switched on or not switched on. So you see this diagram here. There's very cold climate on the left side, all the way until hot climate. You can see that it's cold, right? The tips of this rabbit, Himalayan rabbit, will be dark in color. Because usually when it's very cold, right, which part of the body will be coldest? You feel your hands, your feet, right? Your ears, your nose tip, right? So these are the tips that will become very cold. So for them, in very cold climate, those are the tips of the body that becomes very cold will cause the dark pigment to be produced and give the dark fur. I think it's because dark fur will absorb heat. So you have to absorb heat in the daytime to maintain the body temperature. So that's why it's dark at the tips there. So it's very interesting adaptation, isn't it, to environment. Next example is inheritance of cob length in maize. So do you know what is cob length? You know the corn that you eat, the whole piece of the corn that you eat, this whole piece here, okay? This is the leaf. Looks like banana. Looks like banana. Okay. You know the whole piece of corn that you eat is called a cob, okay, the cob. Now they say that cob length is also coded by allele, just like our height, right? So there are two different kind of phenotypes here. You've got black Mexican and tom tam. So they say that the genotype of black Mexican is AABB and tom tam is small a, small b, small b. So they are all different in genotype. So when they go to the field and randomly, let's say they randomly pick, say 10, 20, 30, 40, 50, 60, 70, 80. Let's say they randomly pick 80 cobs, 80 cobs, and then they measure the length of the cob. Then they find that for black Mexican parent, okay, black Mexican. Now, black Mexican, all of them have got same genotype, right? You see? All are big A, big A, big B, big B. But when they measure the length of the cob, they realize that it's not one length. It's actually a series of lengths here. Okay? So that means to say that there are 3 cobs out of 80 that are 13 in cm. And then there are 11 cobs that they found that is 14 in cm. Then 12 cobs they found is 15 in cm, and so on. So they find that even though all the parents have got the same genotype, but how come the cob length, that means the phenotype, is different? So that means it must be due to environmental factor. Like for example, this plant can get more sunlight. Or this plant can get more nutrients. That plant has got infection, so they cannot grow so well. So because of the environmental factor that's different, it gives rise to a different phenotype, even though the genotypes are all the same. So this will illustrate this formula here, right? VP is equals to VG plus VE. If VG is the same, how come VP is different? That means VE is different. Okay, so if this is the same, if all of them are the same, how come this one is different? Then it's because of this one that's different. Question mark equals to question mark. Okay, right. In the same way, we examine the second type of parent, which is tom tam. So tom tam, again, all of the parent plant have got same genotype, small a, small b, small b. But when they measure the length of the cob, they find that actually it's not one type of length. It varies from 5 cm to 8 cm. So again, the variation is due to an environmental factor. And then when the two parents cross, we expect all the offspring to be heterozygous, right? That means the offspring must be AABB. You can write this into your notes. So if all offspring got same genotype, again, we expect all the phenotype must be exactly the same, right? But now we see, again, there's a range of phenotype for cob length. So this range means variation in the phenotype is due to variation in environmental factor because genotype is the same, right? It must be environmental factor. Now, when they take F1 offsprings and then cross among them to produce F2 offsprings, you learn already from your genetic diagram that the offsprings you're going to get, the phenotypic ratio is 9, 3, 3, 1, isn't it? 9, 3, 3, 1. Whereby 9 means their genotype is A-B-, 3 is A-BB, 3 is small a, big B dash, and the 1 is small a, small b, small b. So you have learned this one already. So based on the genotype alone, we say that there should be 4 different lengths of cob, isn't it? But again, when they go to the field and do sampling, they realize that actually it's not 4 different types. is actually a range of them that ranges from here to here, meaning ranges from 7 cm all the way to 19 cm. So again, there's not only variation in genotype, there's also variation in phenotype in the F2 offsprings. Okay, so the F2 offspring, you can illustrate that the variation in the phenotype is equals to variation in the genotype plus environmental factors. So this is a very good example to show this formula. So let's look at other examples, which is quite interesting. This one all of us know, right? You're born with fair skin, you go under the sun, you become very dark. It depends how long exposed to the sun. And so you go snorkeling, right? You go snorkeling every day, go a morning, go for 1 round snorkeling, then have lunch, rest of our afternoon, go for another round. The 3 days or so like that, after that you come back, totally dark already. Yeah? So why? Because skin color gene is light, but then plus exposure to UV light from environment give you your final phenotype. So excessive melanin production, right? Same as for fruit color gene. Fruit color gene, they will only be switched on at certain wavelength of light, okay, to give you the final fruit color. So from green to red, or green to yellow, or green to orange, okay? And this one also same thing. Now you already learned that bacteria with lact or prawn, they will only switch on the gene when lactose is present, right? Because only when lactose is present, they need those enzymes coded by the gene to metabolize the lactose. So they will only switch on the gene when the lactose is present. So that's why the bacteria may be carrying the lactase gene, but only when there's presence of lactose that the lactase gene will be synthesized. So the phenotype, the final phenotype, that means whether the enzyme is present or not, is dependent on both the genotype, so is there a gene for that enzyme, plus environmental factor, whether lactose is there or not. So that determines the final phenotype. Okay. Now, last one is very interesting one. This one tells us that what we eat is what we are. Okay, a very good example. See, for bees, honey bees, fertilized eggs will become female. Okay, fertilized eggs will become female. So this female larva hatch already, right? Hatch already. If they are being fed with royal jelly only for 4 days, they will become the queen. But if they are fed royal jelly for 3 days only let 1 day, 4th day onwards, they are fed with pollen nectar, they become the female worker. Just a difference of the food by 1 day, totally become different. So imagine what food we eat every day. What do we eat? It will affect your body, you know, okay? Yeah. And then when it affects your body, affect your mental state, right? Let's say if you're very alert, you're very fresh, you know, then after that you can focus very well, and then your emotion is also more balanced. Then the quality of life becomes better. Everything you do, when you meet with people, you talk to people, it becomes better, right? As compared to every day eat like McDonald's, you know, wow. Then after that you feel like very lethargy, you know, you cannot focus, you cannot wake up in the morning. After that, it's like a whole day, every day like that. Wow. Can you imagine quality of life? How you mingle with people, how you go and do things is so different. So the food is important actually. Try to take food that is still in a form that you can see. Like if you eat fish, you can see the bone is better than you can see fish bone only. It's processed. If you eat wheat, it's better to see the wheat grain than to see the biscuit. Highly processed. Correct. Okay. So if you can see the original, the more original, the better. Because the more processed, the less nutrients, the more chemicals added. Okay. Unfertilized egg. All unfertilized eggs becomes male larva, all destined to become worker. So therefore, only feed royal jelly for 3 days enough. Okay. Right. So interesting, right? So this is a past year essay question to ask students to explain using examples, means a few, how the environment may affect the phenotype of organism. 8 marks. So last time essay question 8 marks. Now in structure question 5 to 6 marks maximum. So they can convert the essay to now structure because now no more essay, right? So they can convert it to maximum 6 marks. Still essay. For me, it's still essay. So it's good to study essay. Yeah? So this one you read on your own can already, okay? Right. So let's recap what you have learned for this lesson is genotype plus environment factor will give us the final phenotype. So in a population, there's a variation in the genotype and variation in environmental factor will give variation in phenotype of every individual in the population. Okay. So genotype is nature, environmental factor is nurture. So VP equals to VG plus VE, where V is variation. So examples we've looked at is human height, Himalayan rabbit fur color, crop length, skin color, lactose metabolism, and the role of honeybee not in syllabus. E is not in syllabus one. Okay. So now we have looked at variation. Let's learn a little bit more about the variation. We know variation exists in population. That means to say that there are different phenotypes or characteristics shown by organisms in a natural population that reproduce sexually. Like in the whole class, everyone looks different, right? So there's variation in the phenotype already, right? In a population, they're sexually produced. Cats, human, hummingbirds, finches, etc. So what are the sources of the variation? So we know that the sources of variation comes from variation in the genotype plus variation in environment. Now, variation in environment we already examined just now. Food, sunlight, presence of lactose, and so on. Now we're going to look at variation among genotypes. Why are there differences? Okay, like this, all different. Why different? Okay, different because we have learned that why sexually producing organisms, their offsprings are all different is because of, this one from another chapter already, right? Meiosis. During meiosis, is crossing over of non-sister chromatids of homologous chromosome during prophase 1. Independent assortment of homologous chromosome during metaphase 1. There's random fusion of gametes, random mating of individuals, and then mutation. All these will give rise to genetic variation of individuals in the population. So among all these, right, among all these methods which can give rise to genetic variation, meiosis all the way until random mating here is actually reshuffling of existing alleles only. Right? For meiosis, you're reshuffling the existing allele between the homologous chromosomes. You reshuffle to create new combination. Right? Random fusion of gametes also reshuffling. Gametes from father and mother to the offspring. Which gamete? So which allele? And bind together to become which zygote. Okay? Random mating, which parents gamete fuse to end up which parents gamete. So all these three are working with existing allele but reshuffling only. Reshuffling into different gametes, reshuffling into different individuals. Okay? Mutation is different. Mutation will give rise to a completely new allele in the population. Originally don't have one. Because of mutation, some mistakes occurs and a new allele appears. Okay, there's no reshuffling for mutation. So this one is a revision. Genetic variation comes from here. So next, so we say that we want to study the two types of variation in the population. It can be continuous versus discontinuous. So look at the graph here. Continuous variation is when on the x-axis is the phenotype. And the phenotype, variation among phenotype is continuous, like human height. You measure human height, right? It's actually continuous one. It's like, this is actually frequency. The frequency means number of people having the height. So majority of people will be having height around the mean value of the height of the population. This one is extremely tall. This one is extremely short. So this continuous variation of phenotype. Whereas discontinuous means what? Discontinuous means it is categoric. Like blood group, you've got A, B, and O only. So it's category. It's not a continuous, like quantitative. So left side is quantitative. Can you see? Right side is qualitative. Okay, quantity versus quality. So example, height or mass is quantitative, it's continuous. Albinism, hemophilia, sickle cell anemia, Huntington disease is either present or absent of disease, right? So present or absent disease is actually qualitative category. Either you have it or you don't have it. Okay. So that's the differences between continuous and discontinuous variation. So this is what we're going to learn in more detail about these two variations. Continuous variation, the phenotype is quantitative like height and mass. And then why? So what codes for this variation? There are many additive genes. Additive genes means every gene code for height. There are many genes in our body that code for height, not only one gene. So every gene code for height, and the effect of the gene is that they add up to one another. Okay, let's say you got 5 genes. Each gene code for, let's say 50 cm. So 5 gene will code for 5 times 50 cm. Okay, so it gives you an additive effect. One gene, let's say it gives you 50 cm. So you got 5 genes. Additive effect means it'll give you 50 times 5 cm. 50 cm times 5. So 250 cm. Oh, so tall now. Okay, example only. Okay, too tall already, this one. Okay. And then we know that genes got different alleles, right? Genes can have different alleles. So different alleles in a single gene have very small effect on the phenotype. Now, because this gene code for what? This gene that codes for height. So the allele of the gene also code for height. So different allele means different height, but the phenotype is still height, right? So the differences between different allele is very different, is very little only, because all of them code for height. Either it's 1 cm taller, 1 cm shorter, and so on. Okay. So the differences between allele are very small. Different genes have same often additive effect. So because all genes code for height, so they have the same effect. They code for height. The only difference is that they code for different heights. Okay. So a large number of genes give combined effect on the phenotype. So whenever you've got a continuous variation for phenotype, usually it's coded by polygenes. Polygenes means many genes are involved coding for the same phenotype. And the effect is additive. And the alleles will have very small effects. Different allele got very small effect on the phenotype. Versus discontinuous variation, which is also called discrete variation. Discrete because the phenotype is discrete and categoric. So the phenotype will be qualitative or categoric, like ABO blood group and all the genetic diseases. And flower color, either it's purple or it's white. Okay. So usually there's only one or a very few genes that will code for the characteristic. One or few genes that code for characteristic. Different alleles for a single gene locus can code for very different phenotype. For example, if you say hemophilia, XH versus XH. It's the same gene, but it codes for different, it's a different allele. It codes for totally different phenotype, right? Because the small h will code for hemophilia. The big H will code for normal, right? So therefore, for discontinuous variation, different allele will give very different effect. Because you know already, mutant allele will give the disease. Normal allele will not give the disease. So difference of allele give very different effect on phenotype. Different genes have very different effects also. If there are a few genes that control it, different genes will give very different effect. So these are the differences of the genetic materials between the continuous variation and discontinuous variation, or the genetic properties, okay, for the phenotype, coding for the phenotype. The differences in the genetic properties coding for the phenotype, which is continuous and discontinuous variation. So let's look at the additive effect of the polygenes and alleles in continuous variation, taking height as an example. Okay, polygene for height of an organism. So let's say parental phenotypes is 4xcm tall and 8xcm tall. So let's say, for example, parent genotype, small a, small a, small b, big A, big A, big B, big B. So let us assume that each of the allele code for 1xcm height. Each of these code for 1x, so 4 of them code for 4x. Okay, so each one code for 1x. 1x. This one here, let's say assume each one code for 2x, so altogether 8x. Okay, so gametes is formed, gametes fuse and form the offspring. So offsprings will be, this is 1x, this is 1x, right? This is 2x. Sorry, this is 2x and this is 2x. So altogether, 6x. So offsprings will be 6x tall. So you can see the effect, additive effect of the polygenes and the alleles to give the final phenotype. So now we continue to cross the F1 phenotype to produce F2 offsprings. Cross the F1 offsprings to form the F2 offsprings. So F2 offsprings, what we will get, you have to use the Punnett square to see the result. So for every Punnett square, remember when you write down the genotype, you will also write down the phenotype, so that after that you can count the total number of phenotypes for each of them. So altogether, we can find here, what do we have here? We have a range of height from 4x cm. This is the smallest one, right? Shortest one is 4x cm until the tallest one is 8x cm. But in between, you got 5, 6, 7. So when they plot the graph, frequency graph of the phenotype based on the F2 offsprings, this is what they get. You see, 4, 5, 6, 7, 8. So this looks like continuous, right? It's a continuous variation. Because 4x also got, 5x also got, 6, 7, 8 also got, is continuous. And it's also normal distribution. Majority will be in the mean value. This is extreme. Okay, and this is extreme. So the question here is, how many category of height are there in the graph? How many? 1, 2, 3, 4, 5. 5, isn't it? So 5. So write down your answer 5. How to increase the number of different types of length category? That means I don't want to have 5. I want to have more than 5 different heights. How can I increase the heights? I can use more genes, right? Because the genes got additive effects. When there are more genes coding for height, then there'll be more different type of heights. I can also use more alleles because different alleles give different height, so it gives additive effect again. Okay, and crossing over of linked genes will create new combination. Let's say, for example, we have got 2 genes, right? Let's say this is a paternal and maternal chromosome. So this is paternal, homologous chromosome, and this is maternal chromosome. Okay, so these are the genes coding for height. Say, for example, 2 of them here. Okay, so this is gene coding for height from mother. So let's say fathers one are all dominant. So we just put A and B lah. Okay, big A and big B for father's allele. And then mother is small a and small b. So because they are homologous chromosome, they can pair up, and crossing over between non-sister chromatids will happen. So when crossing over occurs, like before crossing over, you know that A and B will always be together, right? So if you receive A and B, it will give you certain kind of height, isn't it? Always. But now because of crossing over, we have got new allelic combinations. We have got AB and AB. These are new allelic combinations. So now this new allelic combination is going to give rise to a new type of height already, which originally don't have. Okay. So that's why they say that crossing over of linked genes also will give you a new combination of allele, which give rise to a new type of phenotype. Why? Okay, let's say, for example, this one gives 2x, very easy. This is 2x. Okay, the dominant one gives you 2x. The recessive gives you 1x. Okay, 1x. So originally, if there's no crossing over, any gamete that receive this chromosome will express 2x plus 2x is 4x, right? Okay. And then any gametes that receive this chromosome will express 1x plus 1x is equals to 2x. But because of crossing over, now we have got offsprings that receive this one. So this is what is the height for AB. 2 plus 1 is 3x already, you know. So totally different from the original one. So the 3X will suddenly appear. Why? Because of crossing over of linked genes. And this one also is 3X already. So originally no 3X one. So crossing over of linked genes can give rise to a new allelic combination, and the additive effect of the genes give rise to a new type of phenotype. Okay, so this is very interesting. Huh? Okay, so let's answer this question. Classify the following statement as continuous or discrete variation. Okay, you all try. Coat color of hamsters coded by codominant allele C for black and allele small c for white. Okay, start from you. Again? Discrete variation, correct or not? Correct. Discrete. It's either black or white, right? Okay, or black or gray color. So discrete. Second one, proportion of male to female children in family of 8. Which one is this next? Continuous, good. How come no continuous? Continuous good or not? Yes. Discrete. Discrete. Hey, continuous is what? You got, this is totally female. This is totally male. And this is gray area. It's not male, it's not female. It's lala. Cannot, ma. Male means male, female means female, right? Okay, so discrete, male or female discrete. Okay, even though they are lala, but then they actually physically is either male or female one, okay? Only the behavior part is different. Okay, BMI of 100 patients with diet-related disease, next. Continuous, yes, because BMI can be continuous. Okay, next. Number of breaths a person makes when running, next. No lah. Hey, discrete. Number of breath a person makes when running. Discrete, man. Oh, not. Number of breath must be 1, 2, 3, 4, 5. So because 1, 2, 3, 4, 5 so it's discrete. I also think so. The answer is D, ma. Okay. Okay, next. Length of fur of Angora rabbits in a population of Angora. Okay, next. Andrea. Continuous. I also don't know. Let's check answer. Yes. It's like your hair length. But for them, they don't cut their hair. For us, we cut our hair. Okay, if you don't cut the hair, also it's continuous. Okay, number of spores produced by a single colony of mole. Next. Continuous. Sure? I also don't know. Hello, number of spores cannot be 1.5 or 1.25. Number of babies, hello? Number of babies, how many? 1.75. Cannot. This is discrete. Interesting, this question. Okay. Discrete means 1, 2, 3, 4, 5, right? Continuous is like 1.48 1.97 correct or not? So you have learned in this lesson everything about variation. First, phenotypic variation is due to genotypic variation plus environmental variation. So VP equals to VG plus VE. Then we learned that there are two main types of variation, which is discontinuous variation and continuous variation for the phenotype. Then we also examine the genetic basis of these two variations. And then don't forget that t-test is also part of the syllabus. Okay. So this is for today's session, not today, this session.